Good morning again. We deal this morning with the vestibular system, which is a fascinating gadget that the central nervous system uses for what we're going to see in a moment, right? And we deal, as usual, with the structure, the function, and its dysfunction in the clinic. First, the objectives that are relatively simple, because we have to understand that the vestibular system maintains the stability of the visual world, and it also the spatial orientation of the body in relation to gravity, and finally it modulates the autonomic nervous system. We have to end up understanding that. Then we have to have an idea of with what and how does it do it. And finally, we should discuss how to interpret the indications of dysfunction of the system that are caused by either peripheral or central reasons. All right. With these objectives in mind, let's start. First of all is a sensory system. So we'll define the types of energy that are in the environment that are particularly suited to excite the receptors of this system. I put here first a definition of the system that more or less resembles the objectives, but maybe it's a little more specific. Because what does it mean, stability of the visual external world? Well, when you move around, the world moves on your retina, as we're going to see eventually in vision, right? And you have to compensate for that, because you would see that anytime you move, the whole world moves. The vestibular system takes care of that by influencing eye movements, as we're going to see. Then you have the story of the body orientation related to the Earth's gravitational field, right? You stand upright, and the vestibular system controls that posture in relation to gravity, and it does that by adjusting muscles, particularly of the axial portion of the organism. And it does that through spinal mechanisms. The spinal cord is going to innervate those muscles, and it is going to be activated in particular ways by the vestibular system so that it can fulfill that kind of role. And finally, you have the modulation of the autonomic nervous system according to body position in space, because it is different, as is readily noticeable, that when you're lying down and you're standing up, suddenly all your blood goes to your lower limbs and your lower abdomen, and you need blood up here, and therefore there must be quick regulations of the blood pressure so that that blood is not pulling in the lower parts of the organism and maintains adequate oxygenation of the upper parts, which are obviously the important ones, right? Now, what we were saying, that I was interrupted by this, is the stimuli. The stimuli in a sensory system is always a form of energy. In this case, is kinetic energy, and within kinetic energy, the precise stimulus for this system is acceleration. So the acceleration can be angular acceleration, like when you move your head side to side, you start the movement and you accelerate the movement in an angular fashion, and so it can be measured as degrees per second in every second, or degrees per second to minus 2. Then there is also linear acceleration in meters per second to the minus 2. And these linear acceleration are of two kinds. One is the linear acceleration that is translational, which means that when you move in a line, or when you are in the car and you suddenly stop, that linear changes in acceleration are sensed by this system. And a similar mechanism is used to take care of the acceleration induced by gravity. All right. Now, once you have the stimulus, you need the second story of the sensory system is the receptor apparatus, all right? And the receptor apparatus is in the inner ear. So the inner ear is carved into bone, dense bone of the temporal bone, and within that cavities of bone, there are membranes, and they form what is called the membranous labyrinth. And in this membranous labyrinth, you have two not compartments, it's a single compartment, but two parts to this membranous labyrinth. One is the cochlear duct that contains the receptors for hearing. We are not dealing with that. And the other part is the vestibular labyrinth. And in that vestibular labyrinth, it has in terms several components, because it has, for instance, little ducts that are called the semicircular canals that we'll discuss in a moment, all right? The semicircular canals are three on each side, one in the anterior plane, one in the posterior plane, and one in the lateral plane, or horizontal plane, it's the same. And then the other portion of the vestibular labyrinth are the otolith organs, which there are two. One is the utricle, and the other is the saccule. All right, so that's your receptor apparatus. We have to deal with each one because the semicircular canals deal with angular acceleration, essentially, whereas the otolith organ deals with linear acceleration. These are really accelerometers that you have it in your head. Now, we'll start with semicircular canals. As I mentioned here, it's important that detail that all components of the entire inner ear, both the vestibular labyrinth and the cochlear duct, are filled with a thickened fluid that is called endolymph, and an important characteristic of this fluid is the high concentration of potassium ions. All right. It's also important to note that all these components of the inner ear, the membranous inner ear, are intercommunicated, so much so that actually the endolymph and its properties, particularly the high potassium, is being pumped by a portion of the cochlear duct, what is called the stria vascularis, and pumps it into the cochlear duct, but the cochlear duct connects to the saccule, and the saccule to the utricle, and the utricle to the semicircular canals, which means that endolymph that has been generated with its properties in the cochlear duct is filling the entire labyrinth, including the vestibular. All right. Now let's discuss the semicircular canals in some more detail. These are, as I said, as it says there, sensors of angular acceleration. We said that repeatedly. So they are oriented in certain planes of space because they are carved in bone and therefore they are rigid. Only when you move your head they change orientation, right? But the correlation between the planes of each canal is fixed. And you have, for instance, here the right side. In the right side, you have an anterior plane, and perpendicular to that, you have a posterior plane. And between the two, at the base, right here, is the horizontal or lateral plane, right? Lateral plane. These are the planes where the canals are located. So, for instance, in the anterior plane, you have a semicircular canal that goes like that. It's in this plane, in the anterior plane. Then in the posterior plane, you have another one that goes like that. And in the lateral plane, you have the last one that goes like that. Now, each one is in almost an orthogonal position with respect to the others. And as this cartoon shows, the canals themselves, the semicircular canal themselves, have, as we are going to see in a moment, a regular opening on the one side, but on the other side of the canal, the size of the canal gets like dilated in something that is called the ampulla, and we are going to see them in a moment. So you see here the relationship of where the receptors are located, the relationship of the ampulla, that the ampulla of the anterior canal and the lateral canal are very close together, whereas the ampulla of the posterior canal is away from those two. Now, this happens on the right side. On the left side will happen just a mirror image of that. And so now you have very clear the fact, I think, that the planes of the right and left side canals are registered, so that the right lateral canal here and the left lateral canal are in the same plane, in the horizontal plane, whereas the right anterior canal is in the same plane as the left posterior canal. So that's one plane, right? And the reverse for the other one. All right, so that's to have clear in your mind really the location of the structures in relation to space. Now, let's see what each canal shows. This is a cartoon of one canal, all right? You have the duct here and the openings to the utricle. This will be the utricle, all right? And this is the semicircular canal, and you see that there is a plane end here. Well, you know, I'm adding this information, which is important too, that what in this diagram, what is yellow is the membrane, and what is endolymph is the green, is what fills the space. Anyway, you see that there is a free ending here to the utricle, a plane end, and then you have a so-called ampullary end or ampulla. What happens is that the epithelium of this membrane here in the ampulla gets thickened, or makes like a mound, right, in several layers, and that mound is called the crista. Now, on the hills, on the sides, on the banks of that hill, all right, is where the receptor cells are located, and we'll put a series of them here. You may see them or not. Yeah, probably you do. And now these are hair cells that have one real cilium, which is the kinocilium, and as we're going to see in a moment. And here is only the kinocilium represented, which means one cell with one cilium. The next important detail is that the endolymph in that area of the ampulla gets thickened, considerably thickened, all right? So much so that they include all these hair cells in this more viscous mass, and it goes to the other wall of the ampulla, so that there is no free circulation of endolymph. This occludes the canal completely between the crista and the cupula. That's what it's called. That's the cupula. They occlude completely the lumen of the canal. Let's see the consequence of that. Let's deal now with these receptor cells first, all right? Now, you have here a hair cell. You know, it's a cell body, it's a nucleus, mitochondria, all kinds of organelles, and they have an accumulation here in this end of synaptic vesicles that contain a neuroactive substance, a neurotransmitter. All right. This cell, as I said before, has one cilium, which is called the kinocilium. And then it has a series of little hairs that are not really cilia. They don't have the centromere at all, and they are just protrusions, very fine protrusions of the cytoplasm, all right? And they are called the stereocilia. Now, you have that at the tips of the stereocilia, and together with the kinocilium, there are strands of some proteinaceous material that are like strands, right? The so-called interciliary strands of material. Also, another detail is that the tip of the stereocilia contain channels, and these are essentially ion channels. Now, outside is the endolymph at this level, right? And that is, as we know, very rich in potassium, as we mentioned before. All right, this is in the resting state. The head didn't move. All right, now, an important consideration, and of course, things will occur with stimuli that will make this exchange of potassium that will come into the cell this way, All right? This is a very peculiar situation, right? Because extracellularly, you have low potassium and high sodium in the rest of the organism. Here you have high potassium outside. But the only way it will work is if the rest of the cell is excluded from that compartment and behaves like any cell with intracellular and extracellular ionic components. And that's accomplished by tight junctions of neighboring cells. The neighboring hair cells have these tight junctions that isolate this compartment of the cell versus this compartment of the cell. All right, the cell bodies contain other channels that are at this moment closed, like this calcium channel, this is a potassium channel, never mind. And then the last thing is that, of course, this hair cell, this receptor cell, is innervated by a nerve fiber from the vestibular nerve, all right? So we have to put here a terminal of the nerve fiber, and here a synapse is established, right? Between the presynaptic hair cell with synaptic vesicles and the postsynaptic thickened membrane on the side of the afferent fiber. Now, let's see what happens when the head moves, all right? Let's say you turn your head. When the head moves, what's going to happen is that the kinocilium will bend in a particular direction, all right? And we'll see the meaning of that. But when the kinocilium bends, it stretches and pulls with it the stereocilia. So the stereocilia also bend, and you see that the interciliary strands get very tight as opposed to being laxed as before. That results in an opening of the potassium channels, as you can see here. And so potassium rushes into the cell. Potassium, the cell gets high potassium, therefore gets depolarized. And it has its own voltage-dependent calcium channels, that when the cell is depolarized, the calcium channel opens, the calcium comes into the cell, and that leads through several steps to the release of the transmitter in the synaptic cleft and the eventual stimulation of the afferent fiber. At the same time, this cell has also a calcium-dependent potassium channel that opens up, and potassium now leaves the cell, and the cell hyperpolarizes. So there is a constant shift from depolarization to hyperpolarization. And that's what is actually the stimulus that the vestibular system requires to do its job. All right, now, if we deal with just the horizontal or lateral canal, which is the easiest way to discuss in terms of being didactic, now we have to think in terms of how do they work. How does this semicircular canal sense that angular acceleration? So, one thing that we said before that is very important of the vestibular system is that influences eye movements. So what happens when you turn your head with the eye movements? So you can do this cute exercise. Ask your neighbor, all right, to look past you, to look at infinitum, past you, all right? And then you ask the person to turn the head to the right and watch the eyes. And what are you going to see? You are going to see that both eyes are going to the left. So essentially, if my eyes are straight now at this point, right, and I turn my head to the right, you see that my eyes are going to the left. This is what is called the vestibulo-ocular reflex, which is a very rapid reflex, has only, let's say 2 synapses on the way. It's a two neuron reflex arc, right? That was initially discovered as such by Sentagotai, that famous guy that I discussed in the cerebellum. All right. Now, at this time, let's see why it happened. You have the empirical fact, all right, that you turn the head to the right and your eyes go to the left. Now, let's see why this happens. All right, you have your two canals, your two horizontal canals in this location, in this situation, and the cupula and the ampulla are here, are on the front. All right, so that's what I'm depicting in that cartoon. On the right side, you have the semicircular canal here with the ampulla forward. And then you have your left semicircular canal also in the same plane, horizontal, with the ampulla also facing forward. Now you turn your head to the right, the whole head. So the whole thing turns to the right. So what's going to happen with the endolymph here? Well, there is inertia, there is all kind of currents in the endolymph, but the end result is that this endolymph is going to move the reverse as the head turning. And you see that that effect produces a push of the cupula. The endolymph pushes the cupula on the right side. But on the left side, the endolymph is doing exactly the same trajectory, but in this case, is pulling from the cupula. And now you can say very clearly that when you record, let's say, from the receptors and from the vestibular nerve fibers, that pushing the cupula results in excitation, whereas pulling from the cupula results in inhibition. Now you put it together with your empirical fact, and you're saying, aha, when I turn my head to the right, my eyes move to the left through the action of this excitation of the right canal and inhibition of the left-hand canal. Right? That you know empirical, you have seen it. Now comes the problem of when you, let's say, you turn your head to the right, all right? You turn your head to the right, like that, and your eyes go to the left. And you cannot, you can keep on turning the head to the right, correct? But to a point. And the eyes can go so much to the left, and that's all. But if you persist with this rotation, you will see that the eyes will jump to the right, and again will deviate to the left. And they jump to the right, and they deviate to the left. That alternating movement of the eyes is called nystagmus. Now, what happens, of course, is that if you would keep on turning the head, and the only example that I have for that is the movie The Exorcist, that you see this girl with a screw head that goes all around. Well, but that's not, it's only in the movie. In here, the end result is that you have this resetting of the eyes to the right and going fast, going slow to the left and jerking back to the right. In a nystagmus that, by the way, is unfortunately named always, no matter what the course of the nystagmus, is named by the direction of the fast phase. So that means that if you turn the head and the body you rotate the whole body to the right, so the eyes will go slowly to the left and fast to the right. That means that nystagmus is called nystagmus to the right, in spite of the fact that the actual origin is the slow deviation to the left produced by the vestibular system. I hope I didn't confuse you much. All right. Now, during the rotation, then there is this alternation of slow eye movements to the left and quick resetting to the right, right? And that's a nystagmus that is called perrotatory, which means the nystagmus that occur during rotation. And this you cannot see unless you record from the eye movements in particular ways, right? But now suppose that after this rotation, this is stopped. The rotation is stopped suddenly. What's going to happen? Stopping the rotation means a sudden maximal deceleration from rotating to stop, right? You will see now that the direction of the nystagmus reverses. And that's the so-called post-rotatory nystagmus that now in this case will be to the left. Now, when the subject is asked to stand up, all right, the subject tends to fall to the right, all right, and may feel vertigo and even nausea, all right? And there is also a pass pointing. If you ask the person to touch your finger, it will pass point to the right. So there is a tendency to go to the right, the same as the slow phase of the nystagmus. Now, at this point, the problem with the rotatory test, which is that, all right, is that evaluates both sides at the same time. But if you want to know a differential condition of one side or the other in the vestibular system, you cannot use the rotatory test because it gives you the same result of excitation of one side, inhibition of the other, with the corresponding results. Well, at this point, if we can do it, again, I will ask somebody to spin me in a chair, and you will look actually at the results of my sudden stopping. Go ahead. Go, 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 go. Go, 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 go. Go, go, go. All right, stop. All right? You are convinced? All right. Now we continue. And the trick here is to try to test one vestibular system at a time, the right or the left. And for that, you use what is called the caloric test. In the caloric test, you irrigate the ear canal, the external ear canal, with water. You irrigate it gently. You take a syringe and you put a rubber tip and you barely put it inside the external canal, and you squirt the water into the external ear. All right. Now you have to be very careful because there are three variables that you have to consider here, and that the variables will determine the response. It depends on what is one or the other, and this is one of the best examples that you have to learn the concept. Because if you learn the concept, you'll be able to answer any particular question on the system. If you try to learn it by heart, a particular response is useless because it's always a 50-50. You have half the chances of being wrong. So you have to really grab the concept and think about the concept before answering. Now the variables are the position of the head, the side irrigated, and the temperature of the water. And any variation of these three factors will change the response. How does it work? Well, we are testing, for the purpose of the teaching, we are testing the horizontal or lateral canals, all right? Which are, after all, when you irrigate the ear, are the ones that are getting most of the, pulling out of the heat and getting cold, right? If it's with cold water. If it's warm water, it would be the reverse. So let's take a case in which you are going to have the patient or the subject just erect or slightly reclined, because when you recline the head a little bit backwards, the canals are really, really straight in the horizontal plane, right? When your head is straight, they are a little inclined. All right, so you have your head erect or reclined. You irrigate the right ear, the right side, and with what? With cold water. What's going to happen? So you have your right semicircular canal, right? With all the components, the same as we discussed before, and you see it from the top, so you see the canal from the top, like that, with the cupula anteriorly, right? And now you squirt cold water. What's going to happen? The endolymph in this general area that receives this cold stimulus, all right, is going to become denser and contract. And by that function establishes convection currents so that the endolymph will pull the cupula away from the utricle. Now we know already that pulling from the cupula means inhibition, correct? That means that in this situation, it is inhibition that is acting on the right side. What will be the result of that? Well, if the right side is inhibited, the predominant one is the left side that will pull the eyes to the right and will reset to the left. So the nystagmus, the resultant caloric nystagmus, it will be a nystagmus to the left. All right? At the same time, if you stand the patient or the subject, it will tend to fall to the right and will be the past pointing also to the right, which is the actual vestibular response. The unfortunate thing is that nystagmus is called to the left because it's named by the direction of the fast phase. So let's get other variables. Let's say you are doing the same thing, reclined, also the right side, but you put warm water instead of cold water. What's going to happen? Well, you have here the canal, the same canal, right? But now you're squirting warm water here. And what's going to happen with the endolymph in that general area? It's going to expand, become lighter also, right? And the convection current will be such that the endolymph is going to push the cupula towards the utricle. And that we know that means excitation of the hair cells under that situation. And what's going to happen? If the right side is excited, the eyes will go to the left and beat rapidly to the right. So the nystagmus actually will be to the right, but the falling of the patient when standing up or the past pointing will be to the left. Well, this is thing that you have to think about it and try to solve various problems. I'll give you two more examples just for the kick of it. For instance, let's say the patient is reclined, the same as before, but it's the left side that is irrigated instead of the right, and is with cold water. Now, we are talking now about the left canal, all right, with the cupula forward. Now you squirt, in that ear, you squirt cold water. What's going to happen with the endolymph here? It's going to contract, and the convection currents will pull the cupula away from the utricle, resulting in inhibition, correct? So where is going to be the eye movements, how it's going to be reflected in the eye movements? Well, if there is inhibition of the left, all right, means that there is a predominance of the right that will pull the eyes to the left, and the eye will reset to the right. So it means that the nystagmus will be to the right, but the past pointing and the falling will be to the left. All right, one last one that is rather cute, but you have to think about it. Suppose you put the patient in the prone position, which means head down. Right? In head down, now your canals are in this plane, and the ampullas are down there. So what's going to happen? Let's see. You have that your, what canal? The right canal. The right canal is, with the head down, you have the cupula down here. Now it has changed the orientation in space because it's fixed to the head. And if you put the head down, this will go down also. And you put now here cold water, or warm water. All right, warm water. What's happening to the endolymph here with warm water? Will become lighter, all right, it ascends. The convection current will be such that it will pull from the cupula far from the utricle. And what does it mean, pulling from the cupula? Means inhibition, correct? That will be inhibition. So you see that the result will be the same as the previous. No, because it will be on the right side. The inhibition is of the right canal, and therefore the left canal predominates and pulls the eyes to the right, and the nystagmus will be to the left. So you have nystagmus to the left and the falling and past pointing to the right. Now, you have to think about it, understand the concept, understand the mechanism, and any question that's being asked varying any of the three variables or all will give you the correct answer. All right. So we finished that way with the semicircular canals. We have to go now to discuss the otolith organs. These are sensors of gravito-inertial acceleration, all right? Gravity in terms of the acceleration due to gravity, and inertia in terms of the translational acceleration when you move backward or forward. Now, the otolith organ, all right, is part of the vestibular labyrinth, as you see. You have the semicircular canal here that we discussed already, and you have this bubble, this first bubble that is the utricle, and then the second bubble that is called the saccule, and the saccule, the cavity is connected to the cochlear duct that will do the 2.5 turns for the cochlea for the sense of hearing. But now we are interested in what happens here at the utricle and the saccule. And you see now that the membrane at the utricle, in this bottom portion of the utricle, gets thickened. Right? And on top of this thickening, you have hair cells. And on top of the hair cells, you have, and the same happens in the saccule, but you see in a different plane. I will discuss in a moment. These patches that are called macula, right? The utricular macula is almost in the horizontal plane, whereas the saccular macula is in the vertical plane. So you have good sensing mechanisms right there. Now, if you look at more closely at one of these macula, like here, for instance, what you are going to see is that you have the epithelium thickened and you have the hair cells protruding from it, all right? And on the tip of the hair cells, the endolymph is thickened and grabs the cell, the hairs actually. But in addition, this thickened endolymph here contains crystals of essentially calcium carbonate. These are called the otoconia. The hair cells are here, the thickened endolymph is here, and the otoconia are there. All right. Now, when you look at one of these macula head-on, for instance, the utricular macula, you see that has this general shape. This is the shape of the utricular macula, see? When you see it head-on, all right? And now you'll see that in one region of this macula, all the hair cells have the kinocilium oriented in the same way towards that center portion. And when you look at hair cells on the other side, the kinocilia also are facing that same line, if you wish. So some kind of a line is established here so that all the kinocilium of the hair cells are looking at that line. That line is called the striaula. So you have here a gadget, a tremendous gadget to sense any kind of different orientation of the head. Not during the movement of the head, just when you change the position of your head. You have your head straight up, and then you decide to put the head this way. Well, this will be very differentially excited, or that way, and so forth and so on. So it senses the static change in the position of the head. So what are these statolytic organ doing? We know about the semicircular canals, that's easy, right? Particularly its influence on eye movements. Here, there are various things that happen with the otolytic organ. First of all, you have ocular counter-rolling. What is ocular counter-rolling? Well, when you turn your head to the right, you see perfectly well your eyes go to the left, correct? Now, suppose what you're doing is now rolling your head to the right and keeping it there. The world has changed, but it has to be compensated, and the world has to be steady. If you move your head to the right, bend your head to the right, the eyes will counter-roll to the left. The problem is you don't see that. Why? You don't see that because your pupils are round, and this is a counter-rolling with a steady position of the eye. It's only the eye that counter-rolls, and you see it's a circle, and you don't see it moving. If you get a cat that has slit pupils, then you see perfectly well the counter-rolling. Right? But that counter-rolling exists, and is absolutely essential for the stability of the world when you change the position of your head. Now, another thing that is important that this system does is what is called the righting reflex. In the righting reflex is everything that occurs in the muscles of the organism to keep it in the normal posture. So what's your normal posture? It's standing up, for instance, all right? Now, suppose I bend, I'm going to fall. All this system is going to right me up. And a very clear example is what happens when you throw a poor cat from the third, fourth floor. You drop it to the, in space. What's going to happen? No matter how you drop that animal, it's going to fall with the forepaws against the floor. The body will orient in space according to the gravitational field. All right, that doesn't, you don't do that neither with cats and even less with humans, unless you are one of these gangs or whatever. But in any case, what is interesting, for instance, when the human falls off the window and doesn't die, let's say from a relatively low window, right, a third floor, maybe a fourth floor, or even a second floor, what will be the damage in that individual? The damage will be in his feet. They will be crushed. The feet will be crushed. Because on the fall, this system is going to straighten up the body according to gravity field. And what is going to hit the ground are the feet. All right? So that's the writing. Now, another thing that this system does is influence very much the sympathetic nervous system, autonomic, and what is called the vestibulosympathetic reflex. And this is a reflex that is induced by shifting posture with regard to gravity again. And that's, I think I mentioned this example, that when you stand up from a lying position, most of the blood goes down, and therefore you have to compensate that by increasing changes in your blood pressure and so forth and so on. That takes care, as we are going to discuss in the autonomic nervous system, by certain receptors that are in the near the carotid bifurcation and in the aortic branch, in the arch of the aorta, that are sensing these changes in blood pressure that will be dropped and pushing it up through various mechanisms that we'll discuss. Well, it so happened that the otolith organ is probably the earliest mechanism to compensate for the dropping of the blood pressure on the standing up position, because it's a phenomenon that occurs with extremely low latency. So it takes much shorter time for the vestibular system to influence the mechanisms for maintaining blood pressure than the baroreceptors in the large blood vessels. All right, that's what the otolith reflexes are all about. So we have discussed already the peripheral portion of the vestibular system, both the semicircular canal, sensors of angular acceleration, and the otolithic organ, which are sensors of gravito-inertial acceleration. We have to go now into the nervous system. And we saw already in the description of the hair cell receptors that they are innervated by the endings of a nerve fiber. That nerve fiber is part of the vestibular nerve. But now we have to go a little deeper into that business. So you have that the so-called first-order sensory neuron in the vestibular ganglion, which is a thickened part of the vestibular nerve within the internal auditory canal. This ganglion is made of bipolar cells, as opposed to the unipolar cells that we are familiar from the dorsal root ganglion and the trigeminal ganglion and so forth and so on. So from this bipolar cell, you have part of the fiber that is coming from the receptors, and then that fiber that goes to the vestibular nuclei or the cerebellum. Both of these structures are receiving direct input from vestibular fibers. Now, it's interesting that this path from the fibers from the receptors, bipolar cell, and the fibers of the vestibular nerve going to the brainstem and the cerebellum, they have some myelinated covers. They have some wraps of myelin around, which at the level of the nerve itself are tight, like in any peripheral nerve, but they are also present wrapping the bipolar cell, as if the entire thing, is just one conductive element. All right, now, if you record from vestibular nerve fibers like this one, let's make a recording, all right, an electrical recording. You have now a time scale here of half a second. And so if you record with the head still, without moving the head or changing its position, you see that this vestibular fiber is discharging at a relatively high rate of the order of 80 per second or so. And that's different than usual. It's not intuitively correct, because at rest the neuron should discharge very little, if any at all, but not at this frequency. But in this case, it's very appropriate for detecting excitation and inhibition, which are the two factors that oscillate in the system. Indeed, when there is excitation of that fiber, there will be an increase in the frequency of response. And when there is inhibition of that fiber, there will be a decrease in the frequency of response of this order of magnitude. Of course, if you have a spontaneous discharge that is low enough, you won't be able to detect the inhibition. And in this way, you detect it perfectly well. All right, that's for the vestibular nerve. Now, once it's in the brain stem, the vestibular nerve will end in a group of nuclei that are what are called the vestibular nuclear complex. All right, I'll show you a section of that you may be familiar from before. This section that has the cerebellum on top with the fastigial nucleus here, right? That's the fastigial nucleus. And then you have this vestibular nuclear complex here. Now, if we put it a little bigger, like this, the outline anyway, you have something like that, all right? And you see that it's divided into several subgroups. So you have a superior vestibular nucleus, a inferior vestibular nucleus, a lateral vestibular nucleus, and a medial vestibular nucleus. In this particular projection, you can see pieces of all of them. But as a rule, if you have a section that is this rostral, you will see only the superior. And if you take a section this caudal, you will see the inferior and the medial. So it's not usual to catch it in such a way that pieces of the four nuclei are apparent. Now, let's discuss inputs to these nuclei. Well, the semicircular canals that you have them here, all right, will send influences that will terminate essentially in the superior and medial vestibular nuclei. That's easy enough. On the other hand, the otolithic organ, the macula of the saccule, and the utricle, right, they will mostly terminate in the inferior nucleus. Everything that I'm saying is not precise, it's not 100%. I'm talking about predominant inputs. And indeed, the predominant inputs to superior and medial are coming from semicircular canals, and the predominant input to the inferior is coming from the otolithic organ. In addition to that, you have an input from the cerebellum to the vestibular nuclei, and we discussed already in the cerebellum the peculiar, unique arrangement in which the cerebellar cortex now, that normally projects to the intrinsic nuclei, projects directly to the lateral vestibular nucleus, exerting a very powerful inhibitory influence, because output of the cortex is Purkinje cell, which is inhibitory, as we discussed before. All right. In addition to that, the fastigial nucleus, we also discussed this in cerebellum, is going to send input to all the vestibular nuclei. So as you can see already, there is a very tight connection between the cerebellum, particularly the medial slabs, the flocculonodular lobe and vermal cortex, and the vestibular nuclei themselves. Initially, we also said that the vestibular nerve some of its fibers may end directly in the cerebellum, in addition to most of the fibers that terminate in the vestibular nuclear complex. All right, now let's see the outputs, the predominant outputs of these nuclei. Again, you have the nuclei on one side and you have the nuclei of the other side, right? A mirror image. Now, what is the main output of the superior and medial? Remember, they're receiving input from the semicircular canals. The output from the superior and medial are to the oculomotor nuclei. And this is what takes care of the vestibulocular reflex, all right? So it's very clear. Now, on the other hand, the lateral vestibular nucleus is going to give rise to the vestibulospinal tract that we also discuss in the cerebellum as a very strongly excitatory pathway for anti-gravity muscles and allows you the erect posture and so forth and so on. They go all through the spinal cord to lower levels, but the medial nucleus is a more complex nucleus in its output because it's also sent not only to the oculomotor nuclei, but also to the spinal cord only at cervical levels. Takes care of movement of the head and so forth. In addition, there is a strong commissural connection between the medial vestibular nuclei. And commissures here have a very important role because all that we have been discussing about the vestibular system, its function, and the way you elicit their activity, they all have to do with the balance between the two sides. So the two sides have to be in constant communication and balance. And that one of the important components of that balance are these commissural connections between the medial vestibular nuclei of both sides. Also, these crossing fibers of the medial vestibular nucleus, they reach also the thalamus, certain nuclei of the thalamus, which in turn, eventually after processing, will project to the cortex, cerebral cortex, in the parietal lobe in the ventral portion. So, although all of these features that we are discussing for the vestibular system are at the unconscious level, they are reflex, you don't realize that they occur, still you have some sense of motion and you have some sense of balance that are already at the conscious level and that require, therefore, the action of the cortex that you obtain through these connections, cross connections of the medial vestibular nucleus. All right. In addition, all these nuclei project to the cerebellum. Remember that in the inputs, I said that all the nuclei receive inputs also from the cerebellum. All right. That's what we are saying here is the reverse is true also, that all of these nuclei, to some extent, project to the cerebellum. Now, let's talk about dysfunction. We went through already the structure and the function, and we go now to dysfunction. What happens when the system is at failure? All right. You have a problem with the vestibular system. One important symptom that you have is vertigo. You know, the world spins around you or gets upside down. All right? And you can get also nausea, you know, some autonomic component, and you can even vomit. In addition, you have spontaneous nystagmus. You know that, you know, when we talk about nystagmus, these alternating slow and fast phases of the jerky eye movements that we elicited by rotation or by caloric stimulation in the normal, but they can be a spontaneous nystagmus in somebody that has a problem with the vestibular system. Right? Also, there may be a head tilt to the ipsilateral side when one labyrinth is at deficit. Now, examples of problems with the vestibular system. Most of the time, in a great percentage of cases, right, the problem is in the peripheral apparatus. They are not in the central nervous system, all right? And there are various diseases that produce this, from infectious, toxic, whatever. But one example that is very characteristic and is very amazing is what is called benign positional paroxystic vertigo. That, you know, I have it from time to time. This is an episode that lasts very briefly, lasts less than, let's say, 40 seconds, the most, all right? In which you have the experience of that the world is moving, right? And the postural adjustments that occur because you feel that the world is moving are extremely powerful. I remember once I had one of these fits and I was thrown on the bed. I was near the bed, I was thrown to the bed. In another occasion, I was at the cafeteria and getting a cup of coffee, and the machines are against the wall and whatever. I take a cup of coffee, I walk away, and I saw that the whole wall was coming on top of me, and I obviously went to the floor. All right? These are actually hallucinations. You know, they are things that do not exist, and that you perceive it that way. What is benign, and most probably, I think almost 100% at this point, is due to the fact that sometimes there are debris from the macula of the otolithic organ, this including some otoconia, all right, that get dislodged from the utricle and they come into a canal. And they act as a stimulus to that canal, usually the posterior canal, all right? And the treatment is to reposition it. It's canalith repositioning, that special maneuvers that an experienced physician can do in the office to remove that little piece of debris that is microscopic from the canal. All right, that's an example of a peripheral problem with the vestibular system. Now, what about a central origin? The main example is what we discussed in cross syndromes, when you have the lateral medullary syndrome with a pica infarct that was taking, giving you sensory pain and temperature loss on the same side of the face and opposite side of the body, and the infarct is larger, it takes vestibular nuclei, and they give you vestibular symptoms, right? Then you have the problem of motion sickness. That's a very interesting and complex problem. There are genetic components to it. There are some people that get it and some people that do not get it under particular conditions. One condition that elicits motion sickness is when you go on a boat in the water, because in the water, the boat rocks and rolls, right? In the two positions, it rocks and rolls. So if you are inside the cabin, for instance, and you don't have a look at the outside world, what's going to happen? That your vestibular system senses these changes of the boat, all right? But your visual system says that you're not moving because the cabin is moving together with you. So you don't see the cabin moving at all. So there is a conflict of sensory inputs between what the vestibular system is telling you and what the visual system is telling you. And therefore, this conflicting information from the visual system and the vestibular system results in this autonomic phenomenon of motion sickness with nausea, vomiting, and so forth and so on. There is also a conflicting information even within the vestibular system between what the otolith organ is sensing and the semicircular canals are sensing. They are sensing different things, and sometimes it becomes conflictual and resulting in motion sickness. Now, just to give you a concise idea of the differences, you have a patient with a vestibular dysfunction. You have a nystagmus, you have vertigo, whatever. And so you have to very quickly know whether the origin is in the ear, in the labyrinth, in the peripheral portion, or is in the central nervous system. It's an important distinction, all right? And there are various things that immediately lead you to one thing or the other. One thing is the duration of the problem. When the problem is in the peripheral organ, the duration is usually, I'm sorry, but this is mouse sickness. All right, the duration in the peripheral origin can disappear. So far we discussed already the structure and function and some of the dysfunction of the vestibular system. But as a last thing, I want to give you some tips to rather rapidly differentiate between vestibular dysfunctions of peripheral origin and vestibular dysfunctions from central nervous system origin. Of course, it makes a big difference whether the problem is in the ear or the problem is in the brain. And in this fashion, we can try to do it. Little table, all right? And we said peripheral is one column and central is the other column. And let's see a few factors. For instance, the duration of the problem. In the peripheral origin, vertigo, nystagmus, anything you want, is usually transient. It has a limited duration. Whereas if the problem is in the central nervous system, it's very persistent. So that's one differentiation. Now comes the problem of the nystagmus itself, the jumping eye movements, all right? When it's of peripheral origin, the nystagmus is usually unidirectional. It's either all the time to the right or to the left, and can have a mixed nature in terms of horizontal versus rotatory also, but it's always in the same direction. It doesn't change, right? So that's an important characteristic. On the other side, on the other hand, the nystagmus of central origin changes with the direction of gaze. So if you look to the right, as the patient looks to the right, it start beating to the right. Look to the left, start beating to the left. All right, it's multidirectional. Mixed horizontal rotatory I already mentioned, and the central origin is usually pure in direction. All right, it depends on gaze, but it's always the same in the particular gaze. Finally, you have the problem of suppression, visual suppression of the nystagmus by fixating on something. All right, the patient has this nystagmus, jumping eye movement, and you said, look at my finger, and the nystagmus stops. That means that there is visual suppression of the nystagmus when it's of peripheral origin. When it's central origin, that does not happen. All right, keeps on going. Cannot be suppressed by visual fixation. The other thing, which is of course very important, is the concomitant phenomena of different other dysfunctions of the peripheral or central origin. For instance, in the peripheral origin, when the problem is in the periphery, in the inner ear, you will have also problems with the cochlear function, auditory function. You may have hearing loss, or you may have tinnitus, you know, this ringing of ears when there is no ringing really, right? And on the other hand, on the central origin, you have many things. Because remember, in the lateral medullary syndrome, you have the cross-sensory deficits, you can have dysphagia, and you know, problems in swallowing, problem in articulation of speech, double vision, ataxia, a slew of symptoms very characteristic of central nervous system. So if you have these differentiations in mind, very rapidly, when a person shows a vestibular dysfunction, you'll be able to lead yourself into a peripheral problem or a central problem. Now, lastly, let's take a few home messages, all right? I'll read it a little bit. The vestibular system maintains the stability of the visual world by driving both eyes in the opposite direction during head movements, that's the VOR, and or steady head position, that's the ocular counter-rolling that I discussed before. Now, remember all the time that the cupula, which is the thickened portion of endolymph, occludes the ampullary end of the semicircular canals and prevents the circulation of the endolymph. The endolymph develops convection current that will either pull or push the cupula, but not doing the whole trajectory into the utricle. Now, remember that at the tip of the stereocilia, you have potassium channels that open when the kinocilium is bent and pulls from the interciliary strands. Now, excitation of hair cells in one canal drives both eyes to the opposite side, that you saw it empirically. In the rotatory test, in which you spin the subject, as we did before, and we stop suddenly the rotating chair, the sitting subject will show nystagmus to the side opposite the rotation. And on standing up, the subject will tend to fall and pass point to the side of the rotation. You know, as a homework, try to figure that out. All right. And finally, you have in the caloric test 3 variables to specify: the side of the irrigation, the temperature of the water, cold or warm, and the position of the head. Cold irrigation of the right ear with the head in the erect position, produces nystagmus to the left, falling and pass pointing to the right. Homework, use any other change of the variables and figure out the results. The last messages, the vestibular nerve fibers show a high frequency of discharge at rest, which is ideal to sample both excitation and inhibition. They terminate in 4 vestibular nuclei and in the cerebellum. The superior and medial nuclei control eye movements, the VOR. The lateral otitis nucleus controls posture through the vestibulospinal tract that exerts powerful excitation on anti-gravity muscles. The inferior nucleus interacts mainly with the cerebellum. The dysfunction is vertigo, nausea, vomiting, spontaneous nystagmus, head tilt. Peripheral origin effects in most cases are transient. Central origin in most cases are persistent and accompanied by other signs of brainstem dysfunction. Thank you very much for your attention. I think you have a lot of material to digest.